Thank you very much for having me here. And, this has been a wonderful time. I've been to Tuscaloosa in the late 1990s. It's great to be back and see all the developments that have happened to the campus. It's pretty phenomenal. So without further ado, thanks Mukesh for the introduction. The title of this talk is Back to Aristotle, Bringing Hydraulics into Plants. This is a topic that is not usually covered in engineering programs, but there is a lot to be learned from plants and how to engineer things, given the fact that plants in general are able to handle stochasticity and variability way better than us engineers today. And so that's the motivation for digging into this topic deeper and hopefully coming out with ideas for thinking about how to design in the future energy transporting systems. Okay, so let's start by some motivation. Plants, as you might guess, are Earth's primary solar energy collectors. If you look at their combined mass, they exceed all other forms of life on the planet. So if you look about the mass per unit area, they have certainly done much better than anybody else. And it's interesting to ask how were they able to achieve this impressive feat? And if you dig a little bit deeper, you discover that they had to rely on physicochemical mechanisms that had to facilitate long-distance transport of water and sugars, basically, as we shall see in this talk. And the reason they had to do that is that the photosynthetic machinery of plants started in aquatic habitats. Plants actually were not born on planet Earth in a desiccating atmosphere. They actually emerged from water. And when they began to populate land, they had to deal with a desiccating atmosphere. And they had to figure out a way to deliver water to their photosynthetic machinery, in a nutshell. So that's an engineering problem that evolution has found a solution for. And it found a solution that somehow is optimal. And what can we learn about this optimal solution that plants have found to move things long distance without expending energy, miraculously. So in this desiccating atmosphere, still the gas exchange that is driving photosynthesis is the amount of CO2 molecules that enter into the leaf surface. And there is a penalty for that. When the plant stomates open, they lose also water vapor. And so they need basically a hydraulic network that takes water from the soil and figures out a way to move it tens of meters up without pumping. And then they have to also move water and sugars down without pumping. Okay? So how do they do that? And that's what we're going to try to answer today. And the idea then is that there must be some sort of an optimization that is going on that requires some minimal energy expenditure. And so since the discussion is going to be about optimal design, you might want to start by asking, what is the connection between optimality and movement? This topic has been a subject of philosophy for a long time. Aristotle, in fact, was the first to point out that motion is caused and can be sensed, and he argued that once motion is initiated, it will continue indefinitely until it is stopped. But the idea is that there is some optimal principle behind motion started actually with Hero of Alexandria in the 10 to 70 AD, where he was the first to propose that the shortest path introduced to explain how light moves, especially reflection, when you have light reflecting on a mirror, it follows the shortest path. He didn't give or offer at least any mathematical justification for it or a cause. There was no causation attributed to the statement. It was an observation. The ideas that mathematized these intuitions, in fact, by Hero of Alexandria started with Pierre de Fermat, who figured out that it is actually the concept of least time. In fact, it's the travel time that is in fact minimized. And he was able to do that by unifying the studies of diffraction and reflection for the first time. In fact, diffraction in a medium is quite different than reflection. And at the same time, just shortly in the same era, Aristotle's doctrines, in fact, were mathematized by Newton's law, at least the first law. And so clearly there is some connection between mathematization, optimization, and how nature seems to work. And we would like to see whether these ideas can be juxtapositioned to plants. Now, the essential breakthrough in physics happened by Pierre-Louis Maupertuis. He was a mathematician. He studied under Bernoulli. And in fact, he proposed the concept of what we today call the principle of least action. This is a modern version. This is not how Maupertuis presented the principle of least action. But basically, his argument is that if you are interested in the trajectory between 2 points that are likely to be taken, say by a ball moving in space, it will be basically minimizing the action, which is the imbalance between the kinetic energy and the potential energy. And if you want to think about a unifying principle in physics, this is about as much as it gets to figuring out a theory of everything. Okay? Whether it be in quantum mechanics, in classical mechanics, the principle of least action seems to apply. But what is more interesting is his assessment of the significance of the principle of least action. And this is a quotation from his work. He stated that the laws of movement and of rest deduced from this principle, being precisely the same as those observed in nature, we can admire the application of it to all phenomena. And then he went on to suggest interesting applications. So he said the movement of animals, the vegetative growth of plants, are only its consequences, and the spectacle of the universe becomes so much grander, so much more beautiful, when one knows that a small number of laws, most widely established, suffice for all movements. And so now, can we do something like this for plants? And so the answer is no, but hey, we are academicians, we're going to give it the college try, okay? Okay, now just to wrap it up, of course, the principle of least action is now much more studied and much more understood, thanks to the work of Joseph Louis Lagrange and William Rowan Hamilton. It led to the discovery of the calculus of variations, and in fact, Newton's laws were recovered from the principle of least actions, using techniques that now we routinely use in physics that are based on the Euler-Lagrange equations. Also, by the way, The Economist and many other optimization principles use the same ideas. So again, now we are going to jump into the plants and see whether we can think about the movement with minimum energy expenditure following similar optimization principles. Now, the idea of water movement in the plants is not new. It started with Stephen Hales. Hales, this is the same Hales, by the way, of the Hale comet, if you are curious whether they're related. Yeah, it's the same person. And in fact, Hale's book on vegetation was in fact encouraged and commissioned by Newton himself. Okay, so this was a major thing. And in it, Hale observes that plants imbibe moisture so vigorously up their fine capillary vessels. These are the vessels at the leaf surface. And he argued that sap vessels are adapted by their exceeding fineness to raise a sap to great heights. In other words, water moving up from the soil to the plant surface, to the leaf surface, seemed to be designed to be like that. And his statement was that, this is observational. Again, he didn't provide any mechanism that quantifies why moisture would move upwards. He was not able to do that. But this was an important observation in vegetable statics in 1727 that then allowed us to start digging deeper into what is happening inside the plant in terms of water movement. And now we have a much better understanding, to jump quickly now to the present state, of what's happening. So basically, if you look at the leaf shown there, what you see in the leaf, at the leaf surface, is that you have these types of stomates. These are basically apertures that enable the leaf to exchange gases with the atmosphere. And basically the two gases we're interested in at the moment are water vapor and CO2. So CO2 enters these stomates. There are CO2 molecules in the atmosphere, so some of them are going to bombard the surface and enter through these stomates, which are really like gateways. And then once they enter into the stomates, they go through photosynthesis, which we will discuss in a minute, and produce sugars. Once these sugars are produced, they have to be translocated down, and they are done, by this translocation is done through a hydraulic system that is called the phloem. And the phloem is really at the edge of the tree, and so basically what happens is that when you introduce sugars in this hydraulic system, you are able to pull water from the adjacent cells by osmosis, and you pressurize the fluid here and you push it down. So that's how actually sugars are moved in the phloem. Now, because you are losing water, so every water molecule that you lose from the leaf, you have to pull this entire column up by one water molecule. And so as the soil adjacent to the roots begins to dry, it takes more and more effort now to pull this entire column up by one molecule. So what happens is that tension begins to develop along the hydraulic pathway connecting the roots all the way to the leaf. So the loss of water here is in fact building tension. And so water now has to move from the soil all the way up to the leaf in under tension. And we are talking about tensions that are not trivial. These are, 3-4 megapascals, okay? Pretty high. And because of that, water is moving in a metastable state. So it is very prone to cavitation. Advantages is that you're not expending energy. This is all done based on certain properties of water. Primarily, it's cohesion. The hydrogen bonds are holding the water molecules together. So you could pull it up. And because of surface tension, when these pores oscillate, shrink or expand, you are changing basically the capillary power to pull that column up. So that's basically, in a nutshell, what's happening. This is kind of how this system looks like if you look at an electron microscope. And the phloem looks somewhat similar, but much, much smaller in terms of volume of water. Now, the talk will argue that in each one of those systems, there is an optimality principle at play. Okay, so that's the summary conclusion of what's going to come. And we're going to try to show that, in fact, these kind of optimality rules that are going to pop up share many things in common. They allow us to describe a certain function with the structure that we see. So let's start with the leaf view. So here's the summary of the optimization principle that is at play. The perception is that these pores that we have been looking at are operating, opening and closure, in a manner that tries to maximize how much CO2 can enter the plant, subject to how much water is available to the plant. So that's an optimization problem. That's, of course, making the assumption that the plants have read the calculus of variations and understood it. Okay, so that's the first thing that one needs to remember. The second idea is that in the xylem, so now this is one of the hydraulic pathways moving water up, is that plants have evolved a variety of strategies to prevent hydraulic failure in their conductive tissue while ensuring safety. So if you have a big pipe, basically, the probability that air enters that big pipe is higher. Okay, so you're more prone to cavitation compared to a much smaller pipe, but a much smaller pipe does not have the same hydraulic efficiency as a big pipe. So that's the trade-off in a nutshell. The third one is related to sugar transport in the phloem, which is where water is moving under positive pressure. And in this case, the argument is that if you pull enough sugars in, well, you increase the driving force for osmosis, but you pay the penalty by increasing the viscosity of the fluid. So there must be some sort of an optimum again there. And so that's basically what we're going to try to show. And then how to bridge all these things together is, of course, the big problem. So let's start with the leaf equations. Now let's we are kind of in the School of Engineering, so we have to show some equations. So let's show some equations. The basic, what the plants want is to photosynthesize, and to do that, they need CO2 molecules from the atmosphere, so CO2 enters here. And they need liquid water for that photosynthesis. And basically what photosynthesis is it's a sugar production and oxygen production. Okay, so this sugar then is what is used to build the leaves, to build roots, to produce fruits for our eating habits, etc. To produce defense mechanisms for the plants, to fight, disease outbreaks, and so forth. And basically, now to use an engineering analogy, since again we are engineers here, the photosynthetic demand is basically linked to how much CO2 concentration there is in the inside of the leaf. So the higher the internal CO2 concentration, which is shown as Ci here, the more flux you could allow to go into this photosynthetic machinery, so you could produce more sugars and more oxygen. Now, the coefficients alpha 1 and alpha 2 that you see here, basically it says which limits this reaction. Is it the amount of photons, the light part, that actually drives a piece of it, or is it the enzyme kinetics that dictate the temperature behavior that are dependent on temperature behavior to produce the sugars and the oxygen? So is it more light limited or temperature limited? Those are going to affect the coefficients alpha 1 and alpha 2 in a known and predictable way. This is basically, if you borrow from engineering, this is nothing but a reservoir problem. You have an outflow, in this case, the biochemical demand, as a function of storage, in this case, the internal CO2 concentration. So you could think of it as a simple nonlinear reservoir. It's a saturation function. As the internal CO2 concentration increases, the photosynthesis saturates to a maximum value. Okay, now this is the demand. Well, where are these CO2 molecules coming from? Well, they're coming from the atmosphere. So as CO2 molecules from the atmosphere hit the leaf surface, some of them will enter, and it is the imbalance between the atmospheric CO2 concentration and the internal CO2 concentration that's going to drive or supply basically the biochemical demand, FC. Now, of course, you have to know how open are these stomates. Okay, what is the porosity of this medium in a nutshell? Okay, that requires some parameterization. So from a mathematical perspective, when you combine these two equations, this one and this one, you basically had one equation initially with two unknowns, the photosynthetic rate and internal CO2 concentration. These were the two unknowns. You had one equation. You introduce basically another equation from physics, basically mass transport, that linked the CO2 flux to the internal CO2 concentration. So now you've closed the original system at the expense of introducing the porosity of this medium or the diffusion properties of these mediums, of how open these stomates are, or the stomatal conductance, as a third unknown. So now to solve, in a nutshell, this problem, you need a third equation. And that's where optimality theory will come handy, is that instead of writing an empirical equation, we can argue that all plants, by and large, might operate using some sort of an optimality rule that will enable us to find a generic third equation that allows us to solve these two equations and two unknowns. So to do that, we might argue the following, that if we envision the carbon gain to the plant as a plus, so that's what the plant would like to do, but they have to pay a penalty where if they open these stomates, they have to lose water vapor. And so that's kind of a penalty to it. And in fact, that penalty is dependent on the same pathway as the CO2 input. So when the stomates open, CO2 goes in, water goes out. And in fact, if we use Dalton's law to describe that water loss, then it will be the conductance of this medium multiplied by the aridity of the atmosphere, encoded here in a so-called vapor pressure deficit. So then the optimizing problem, or the optimization problem, would have this objective: to maximize the carbon gain, which is the photosynthesis, integrated over a time period from 0 to t, okay, subject to a constraint that there is enough water to be able to run this optimization. So that's a constrained optimization. And thanks to the work of Hamilton and Lagrange and Euler, we can certainly convert this constrained optimization to an unconstrained optimization. How? Well, the Euler-Lagrange equations. So we can define a Hamilton, the Hamiltonian that defines the carbon gain and the water loss. And basically, we impose these constraints through a Lagrange multiplier. So that's basically converting, if you like, the water economy to the carbon economy of the plant through this Hamiltonian. And then we solve this optimization problem, this unconstrained optimization problem, using the Euler-Lagrange equation, where the time rate of change of the Hamiltonian with respect to the control variable, here GS, which is the aperture of the stomates, its variation in time, minus the time derivative of the Hamiltonian with respect to the conductance. So these are just the standard Euler-Lagrange equation. But in fact, they get simplified more because the equations themselves don't depend on the derivative of the opening and the closure of the stomates. The velocity of opening and closure is not too crucial. And so you're left basically with one equation that says dH dGs must be 0 because the Hamiltonian is not a function of the derivative of the stomatal aperture. So it doesn't depend on how the velocity of opening and closure is. And remember, these are partial derivatives. So in this case, we can add this condition, as a third equation to our biochemical demand and atmospheric supply of CO2. And notice in this case, we have written the carbon gain as a function of this control variable and the water loss as a function of this control variable. So this condition should tell us what is the optimum aperture that you need to open to maximize your carbon gain given the amount of water you have. Okay, so this is just a statement to say that now we have one equation with two unknown. That was our biochemical demand. We added an atmospheric supply, so we closed the original problem that linked photosynthesis to internal CO2 concentration at the expense of needing to describe the stomatal conductance. Then we imposed an optimality hypothesis that says you need to maximize the Hamiltonian with respect to the control variable GS. So that gives us a third equation that now allows us to solve this system as three equations and three unknowns. And of course, for that, we need to quantify what is the Lagrange multiplier that is often called the marginal use efficiency. And as all optimality principles do, is that you use the constraint to evaluate the Lagrange multiplier. So in that sense, the problem is mathematically closed, and it can yield solutions to this coupled photosynthesis water system. And just to show you how they look like, I thought if we linearize this biochemical demand, so this is FC now is linear in CI, we keep the mass transport the same, we impose the optimality principle in this case and solve these equations, and you end up with these very simplified expressions. And what I want to draw attention to is that if you divide this expression by this expression, you end up with a relation that looks like this, that the stomatal conductance, which was our big unknown, is linearly related to photosynthesis, normalized by atmospheric CO2, multiplied by the vapor pressure deficit. The origin of that term was Dalton's law, to the minus 1/2. So there are two interesting things to point out, is that the relation between stomatal conductance and the vapor pressure deficit turns out to be a power law, and the exponent of that power law is an emergent property of the optimization. It was not empirically imposed. This came out from the analysis. And there is a linearity between stomatal conductance and photosynthesis normalized by CA. And I should say that if you look at the literature, there is plenty of literature to support the linearity between stomatal conductance and photosynthesis over CA. These were empirical observations. And there was lots of observations to support that the stomatal conductance ought to decline with increasing aridity in the atmosphere. And in this case, it's predicted to be a d to the minus 1/2. So is there any support for that? The answer is yes. My colleague Sari Palmroth did a lot of work on Scott spine, where she actually sampled gas exchange. So you clamp the needles and you measure basically CO2 and water vapor across a climatic gradient. And what she showed is that if you plot the transpiration rate, which is the stomatal conductance times the vapor pressure deficit, and you plot that against photosynthesis, which is shown here, so you plot this quantity against E, the transpiration rate, the data seems to collapse on a single line, suggesting that, in fact, the d to the plus 1/2 prediction for the transpiration or the d to the minus 1/2 prediction for the stomatal conductance is correct. So this is somewhat evidence that for the same species, you are able to predict transpiration and photosynthesis and stomatal conductance from this system of equations. Okay. And just to sum it up, the fact that the vapor pressure deficit was important for stomates has been known for quite some time. In fact, Francis Darwin, who, by the way, is the son of the famous Charles Darwin, was also a biologist, was very much interested in plants. And he wrote in a 1898 paper, a paper called Observations on Stomata. And he noted that when we remember the innumerable adaptations which serve to economize water, it is inconceivable that the closure of the stomata should not cooperate. Sorry about the multiple negatives here. That's how, at that time, the writing style was. This is not how we write today, but you know. And in fact, what has just happened from optimality theory is almost a mathematization of the stomatal conductance vapor pressure deficit relation. Okay, so this is all I want to say for now about the leaf. There are many other aspects of this optimality principles that can be explored. We've assumed the Lagrange multiplier is constant. Who says so? There are lots of other features about the optimality principles that need to be adjusted, but you get the flavor of this approach. So now that we have figured out how to describe optimally water movement between the leaf and the atmosphere, now let's talk about the water delivery system that goes from the soil to the leaf. And there is more, how should we say, colorful debates that science has experienced. And the first one was basically what drives water movement from the soil to the leaf. Okay? And in the early 1800s, there were two camps that attempted to answer this. One camp was labeled as the vitalists. That was the technical term given to this group. And the other camp was the physicists. And so the vitalists were convinced that the water movement from the soil to the leaf must involve living cells. Okay? But they were not able to identify where those cells are and precisely how they do it. Okay? So they were just convinced that it has to be a living entity that is moving water. Okay? On the other hand, the physicists were the exact opposite. They were convinced that only dead cells are involved, and hence the mechanism of water transport in the xylem must rely directly on physical effects. No cells doing any jiggling or something like that to move the water. Okay, so in 1891, a famous experiment by a plant biologist, Edward Strasburger, did the definitive experiment to tease apart which hypothesis is correct. And the experiment was beautiful that it is worth repeating its main ingredients here. He cleverly decided that, well, if living cells are involved, can I still sustain water movement in the xylem of the tree when I kill them? Okay? And the way he did this is he actually put the tree in copper sulfate. Copper sulfate is very poisonous for cells. And so after 14 days, all the cells basically must have died because he saw copper sulfate up to the top of the tree, but still found 30 liters of water are still transpiring. So he didn't see transpiration, in fact, stopping much, but in fact continued despite the fact that all the living cells have been killed. So he concluded that transpiration, or the water movement from this reservoir all the way up to the leaf, cannot be involving living cells. He just poisoned them for all practical purposes. He also made some notes that the physical forces are not clearly known at that time, but he excluded barometric pressure or root pressure. Okay, so he was able to exclude barometric pressure because he did this experiment more than once and found that the barometric pressure is not playing a role. And since this was immersed in saturated water, the root pressure was under saturation. Okay, so the final version of our understanding of how water moves in the xylem of the trees came from the Irish duo, Joly and Dixon, and in fact they have met Strasburger before, so they were fully aware of his findings, and they did a much cleverer experiment, in fact, than he did. And that was kind of what nailed the result that they were on the right track to think about capillary forces and surface tension moving water. So what they did is they took a branch and they immersed it in liquid water, and then this whole thing is sealed. Now, of course, you would see condensation if there is transpiration. So what they thought they could do is that if they add positive pressure, so they keep increasing the pressure in the atmosphere to block basically transpiration, they should be able to, if the pressure was negative, in this case they are forcing the pressure of the air to stop the transpiration, how much pressure do they need to stop the condensation on the sides of the wall? And they found up to 3 megapascals are needed to stop basically the transpiration from proceeding. Okay? So they knew that the pressure was negative, and they knew that water was going up under tension based on a positive pressure result, believe it or not. That was the beauty and the elegance of this experiment. So they published the result and proposed basically what we now call the cohesion-tension theory in its final form, and it was highly criticized by Francis Darwin himself. He stated also that believing in the ability of water to bear tension is to some of us equivalent to believing in ropes of sand. So he envisioned the fact that this water column is moving up under tension is nonsense. And of course, we now know from fluid mechanics that yes, Darwin is correct that fluids cannot withstand shear. They can withstand compression, pretty well. In fact, water is sometimes treated as an incompressible fluid. But they can also withstand tension, and that is thanks to the hydrogen bonds and the van der Waals forces that are holding these water molecules together. And as happens in academia again and again and again, what Darwin was not aware of is that there were experiments by Berthelot and Jager that showed that, in fact, water can withstand high tensional forces. So he was just not aware of these studies, even though they were done and published in journals that he would have been familiar with. So while sure, fluids cannot withstand shear, they can withstand tension. Later, after Jager published these results, which showed that water can easily withstand tension of 50 megapascals, not 3 or 4, 50 megapascals. Some questioned the validity of these results by claiming that perhaps what Jager and Berthelot measured is actually adhesion, not cohesion. Okay, so adhesion of the water to the sides of the channel or the compartment, not the cohesive forces, but still the analysis was redone, and it was convincing enough to proceed with the fact that it was surface tension and cohesive forces that held the water together. So after that, everything was going well for the Irish duo, Dixon and Joly, until the Austrian forester Haberland in 1914 published a paper that speculated that there must be some amount of water and air in the xylem. Okay? And so he gave some rationale for why there may have been air in it. Well, I will not go through them all, but this was in his 1914 book. And he argued that vessels and tracheals normally contain both air and water, the relative amounts of the two substances varying according to the season and the time of day. So he was convinced that somehow air must have been there. And it took until 1966 to prove, in fact, Haberland's arguments. And this was done by an Australian scientist by the name of John Milburn, a beautiful paper, and in fact a beautiful lecture that was called Listen to the Trees. What Milburn did was a very clever experiment. He put high-resolution microphones around the base of the tree and was able to listen to the cavitation that was happening during the bubble formation and the bubble collapse. So he was able to literally listen to cavitation happening in the tree. So this was 1966, and John Milburn's work seemed to suggest that formation of air is not an uncommon feature. It's happening all the time, and that was a surprise to the cohesion-tension theory that was envisioning water being pulled up as a single column all the way from the soil to the leaf. Okay, so now what? Now these studies became somewhat popular, and there is a lot of interest in them, simply due to the fact that now we have a lot of droughts, and forest mortality has been associated to droughts and severity of the droughts and the duration of the droughts. If you look across long-term ecological stations that have been reporting droughts and mortality, there is clear connections that more droughts are causing higher mortality rates across the world. And that actually brought up the topics of eco-hydrology and plant hydraulics back into focus as to try to offer explanations how to connect droughts to the survival of forests. And one way to proceed with this analysis is to argue that plant responses to droughts might be analyzed by how much you lose hydraulic conductance when you increase the tension in the water column. Okay, so you don't care about a specific location or a specific member. You are looking at the entire water column and you ask, well, if the column becomes more and more negative, how much loss of xylem conductance I incur? And it was very clear from experiments reported by John Sperry and others that you have typical responses that might go like this: a little bit of tension, no loss in xylem conductance, and then suddenly a very rapid loss in xylem conductance. And those shapes tend to coincide with riparian trees. On the other hand, if you look at desert shrubs, you could go to 4 megapascals and hardly there is a % loss in xylem, and then there is a gradual basically reduction in xylem conductance as a function of pressure. And some measurements even report 12 megapascals before you do that. Now, this vulnerability curve sometimes is referred to as PLC, % loss in xylem conductance, and it has some resemblance to the hydraulic conductivity function in soil physics. So if you remember in soil physics, in unsaturated flow, if you recall Darcy's law, there is an unsaturated hydraulic conductivity that varies with soil moisture or soil pressure. And of course, the drier the soil becomes, the less conductive it is. So these ideas are around, they've been around in hydrology for a while, and there the holy grail in soil physics has been the following. If I give you the pore size distribution or the particle size distribution of soils, can you predict the hydraulic conductivity of that soil? Okay, so that was one topic that soil physics has been trying to solve for a while. And naively, the approach that I've taken is that, well, I know in physics that percolation theory, specifically directed percolation theory, did offer some solutions to this problem. So I thought, well, why not trees? Okay, perhaps percolation theory can link, therefore, the anatomical properties of the xylem, its radius, its length, all these configurations, just like the pores in soils. It might be able to link the anatomical properties of wood to hydraulic properties and hence answer what species resist, if you will, droughts, if we envision the resistance to drought as being somewhat like a desert shrub or the lack of resistance as being as a riparian tree. So this was the original line of attack from a theoretical perspective. And as usual with those ideas, nice idea, but wrong. Okay, and I'll tell you why in a minute, despite the fact that I was the one who was pushing my students to do it. Okay. Don't you hate it when that happens? You have a very good idea, a very nice story, and then the students prove you wrong. Okay, so now just to show you why the advisor was intrigued by this idea is that if you look at the wood anatomy, you find that roughly speaking, the wood anatomical traits seem to cluster in two groups. There is what is called ring porous wood, and there is what is called diffuse porous wood. So what are those? The ring porous wood, these are character. So this is every year the tree grows, and so this is a new year. So, and then up there's a new year. And you see that at the beginning of the growing season, the pores appear to be pretty big. And then as the growing season proceeds to the summer, the new pores that are being put, because the tree is growing, so we're putting new xylem, seem to be smaller and smaller. And then we reach the end of the season. Then the next year you start, and then the pores are big, and then they become smaller. Now, if you look at the vulnerability curve, again, this is a plot of xylem pressure against percent loss of conductance. You find that for this system, it looks like the riparian vegetation. A little bit of increase in xylem pressure, you lose very quickly the hydraulic conductance because you cavitate basically all these big tubes. Okay, they become not conductive. On the other hand, if you look at the diffuse pores, so this is one season, this is the next season, this is the next season, you find that there is kind of small pores across the entire growing season. So there is no preferential structure to them, and in fact they do handle much better stress. So in other words, you could push the system to minus 1.5 megapascals, and then you start losing conductance as the pressure becomes more and more negative. So clearly this is more advantageous in water stress conditions. This is definitely more competitive when water is available because it's very efficient. Okay, so this was the basis that perhaps there is a connection between wood anatomical traits and the percent loss in conductance. Now let's see the physics that has to be resolved and what it shares with porous medium. The problem with water movement from the soil all the way to the leaf is just the multi-scaled nature of the problem, which is usually a killer. You have basically, at the tree scale, well, you have the tree height. Then what happens is that the xylem itself is not a single column. It is composed of either vessel members or what are called tracheals. And these elements are in fact connected, in the case of tracheals here, or in the case of vessel elements here, by membranes. And these tubes are highly anisotropic. So the length of these tubes is like, centimeters, and the diameters is like micrometers. Okay, so very anisotropic, slender tubes. And this connection, this membrane connection here, is in fact has what we call a pit field. And at the nanoscale, this is how the pit field looks like. So clearly, the pores that are allowing, for example, the air to be stopped, in this case, which is the reason why it is designed like this, are really tiny. And so what happens is that as cavitation happens, bubbles form, they become unstable, they block, for example, one of those members, but there is redundancy in the system, so water can move around the cavitated vessels. So even though you cavitate and you lose hydraulic, so to speak, conductance in one of those elements, you have a network of them that are connected, and they are connected, they are connected like this. So these are the typical tubes that you see, and between them are those, kind of membranes that could be approximated, if you will, as thin sheets with holes in them. Now, there are two physics involved as water moves, in this hydraulic network. The first one is the longitudinal, so water is moving up, and there you could argue that wall friction is the dominant mechanism that is dissipating energy, okay? And so you get the Hagen-Poiseuille equation. The pressure drop is related to the flow rate, the radius, this big radius to the fourth, the length of this tube, the viscosity of the fluid, etc. But there is another physics involved, which is more in this direction, and that's actually given by what we call a Sampson flow, where if you have a sheet with a hole of radius rp, you ask, if I impose a pressure difference across this hole, how much flow rate can I pass? And it turns out to be proportional to the cube of this radius. Has nothing to do with wall friction. Okay, that's all I will say for now. And the interesting thing is that the head losses here and here turn out to be comparable when integrated across the entire path. And that means that the tree has built, or engineered actually, a fantastic system. There are no bottlenecks yet. In the xylem, you have head losses. Across the membranes, you have head losses, but they're comparable. There is no one that is so dominant that the head losses in the longitudinal directions are so much bigger than the head losses that you encounter across the membranes. And that's why sometimes in operational models that describe water movement, they just focus on the Hagen-Poiseuille version of this problem. Nonetheless, this is basically for water movement. Let's look at air, because at the end of the day, air cavitating is a problem. Air can enter into the xylem through multiple pathways. You have cracks in the tree. You have scientists drilling holes to put sensors in the tree, so that creates also, air pathways, etc. So you have a lot of those. So once air forms, if you are under tension, that air bubble usually is unstable, and it then fills one of those members. So typically what you could have is you have a functional vessel, you have an embolized vessel, and you have this membrane in between that is kind of short. And so what happens is that the membrane, if it is zero, it's kind of closed. And then what happens is that as you start increasing the tension here, this meniscus, in fact, begins to shift this way. And then if it opens enough, you could have air leaking to the water side. And that air bubble becomes unstable, and it creates basically another cavitation event that embolizes this vessel. And this is very much like how viruses spread in society. Yeah? I mean, this is an epidemiological problem that the trees had to confront, the spread of air in their xylem. And of course, the spread of air happens very much like disease infection. You need an interaction between an embolized vessel with air and the functional vessel with water. So you could think of this as a susceptible and this as an infected person, and it is when they interact together that disease spread. Air spread is happening exactly the same way, okay? Which is another interesting feature about studying plants. Maybe they will teach us something about epidemiology and prevention of disease spreads by localizing and isolating. That's exactly what the tree is doing, by the way, with air, okay? So that's how air is spreading in the system. So to test these ideas as a network problem, what we decided to do is take a real arrangement of these vessels and tracheals, and basically what we did is we imaged them, and we decided to try to preserve basically the length and the diameters and the connectivity of these pipes, but in 2D, okay? So, and that's kind of a maple species that is amenable to letting us do that. And that's the model in a nutshell. This is a highly simplified view of the problem. So you have vessels that have different lengths. These lengths, the distribution of these lengths is preserved by the real data, if you will. And the connectivity between them is also preserved. And of course, the longer the tube is, the bigger the diameter and more connections it will have. I mean, that's almost by virtue of construct. And so now we just assume for the purposes of water movement, all these vessels are vertical. They are initially all water-filled conduits. And then what we did is we had these membranes between them. And the membranes would look something like that, where in fact air spread will be determined by the biggest diameter and the thickness of this membrane, because that is basically giving this membrane rigidity. And so what you do next is you at random introduce air pockets, and instead of supplying the water with tension, we do what Dixon and Joly did. We increase the air pressure. So that's instead of making water under tension, we make the air positive pressure instead. And we study the evolution of water and air in this network. The argument is that if air can go from an embolized conduit to a water-filled conduit, it will immediately render it as incapable of transporting water. And so what we did next is we ran the calculations, and that's a movie showing the pressure of the air after random seeding of air molecules in this network. And what you see is the cavitated vessels in blue. So these are the ones that now air has gone into them and they became unstable. Isolated vessels are shown in the pink. By isolated vessels, I mean vessels that are still retaining their water, so they don't have any air, but they become hydraulically disconnected from the network. Okay, so they don't contribute to the flow from the bottom to the top, let's say. The functional vessels are shown in black, and then the contact walls are shown in red. And so you could see as the air pressure goes up, more and more of the functional vessels become cavitated, but there are big chunks in this network that become hydraulically isolated, meaning there is water in them, but they're not contributing to the overall hydraulic conductivity of this network. And if you compare the predicted percent loss in conductance on the y-axis as a function of the applied pressure on the x-axis, you find that because these were done as Monte Carlo simulations, that basically you have the kind of vulnerability curve reproduced. And the only input to these calculations is really the vulnerability, the anatomical properties that are then used to predict the vulnerability of this curve. So this was kind of encouraging that this problem can be done, but the failure of directed percolation happens because of two things. First, the axial physics is different than the radial physics. The laws that are describing head losses in the axial direction and the radial direction are different. That's not the case in porous media. Okay, so that's the first thing. And then the second thing is that the air spread is very different than the water movement. So they are not as complementary. And just to make that point clearer, this is the pressure drop as a function of percent loss in conductivity. And this is just to show you that, yes, the hydraulic conductivity is decreased with increasing air pressure. But the other thing that was interesting is to keep track of the relative conduit water content. So even at 4 megapascals, when there was complete loss of hydraulic conductivity, there was plenty of relative conduit water content. And this is coming from the isolated network pieces that still retained a lot of water. And by the way, the reason this is somewhat interesting is that now we have, from remote sensing, basically, the ability to measure changes in the relative conduit water content from space, which is pretty impressive. Okay, the last optimality problem, and then I will stop here, we're almost done, which is the sugar mass transport in the phloem. Quickly, this is the phloem, and so if you imagine that the sugar flux is purely advective, so that would be a flow rate multiplied by the concentration of sugar multiplied by the density, so that's the total mass flux here. But the flow rate depends on the pressure differential divided by the viscosity times some geometric function. So if you put this back in here and you approximate the pressure by some hydrostatic form and some osmotic form, given even linearly by the Van 't Hoff equation, you find that if you increase the concentration, you increase the driving force, you increase the viscosity, and you could potentially change the pressure. So clearly there must be an optimal, okay? Because the viscosity, think about maple syrup, right? It's very viscous fluid, full of sugars. Okay, so if you do that, you again find that if you plot the flux on the y-axis against the sugar concentration on the x-axis, clearly there must be some maximum occurring that enables efficient transport of mass. And interesting enough, the numbers that are popping up from this highly simplified analysis seems to be within the range of the data that has been proposed in terms of measuring sugar content in weight, per weight. So that was kind of encouraging to also show that the phloem is operating under optimality. So now some specific conclusions. Well, we have shown that there is an optimization principle operating in every compartment, the leaf, the xylem, and the phloem. However, we have yet to find a general optimality theory that welds all these pieces together. Maybe it doesn't exist, but that would be a goal to think about. There is now in physics a whole literature on networks that's popping up in many engineering fields. So for example, the electrical grid, the telecommunication grid, the internet grid, the telephone lines. I mean, they're all networks on networks. You have a failure in one that has a cascading effects, if you like, on all the others. Same thing. I mean, if you have flooding, you could not only flood, but you could wipe out your electrical grid. So again, similar ideas. And so the question is, can we learn from these mechanisms that have been proposed by Eugene Stanley from the start to deal with this multiple, so to speak, networks involved in water movement? We have also shown that it is possible to predict the shape of the vulnerability curve and relative water conduit from wood anatomical traits. This is very much like a soil physics problem, but it requires more multi-scale processes. And now we are at a phase where we can image a lot of these multi-scale processes. So X-ray tomography, MRI, etc., are now allowing us to see, in fact, in unprecedented resolution, the vessel network in the trees, and perhaps, therefore, enabling new theories to be developed to tackle water movement in the trees. Thank you, and I'll stop here.